So thanks a million for inviting me. This has been a wonderful meeting, and I feel very privileged meeting you all. So thank you. So I recently moved back to Ireland from Boston, where I'd been for the past 16 years. And I think the most immediate thing that kind of struck me is the number of rainbows, which are multiple per day. So hence this slide. And I've come back to to help start up a new institute in the University of Limerick, which is a small university and a relatively new university in the southwest of Ireland. And it kind of might seem like an odd choice, like why didn't I go to Dublin or to someplace else? And part of the reason is it was a technical university that was founded basically by engineers and computer scientists. So it's actually got like all of Ireland's flagship programs in data science and software engineering, and it kind of runs all of these programs. So when I was thinking of where to move to, I want to go where I'm going to be getting the undergrads and the students in, where we can actually build something. So that's very much why I've moved there, in addition to it being close to where my family are from. So I'm going to kind of talk a little bit today. All the work that I'm going to talk today is very much about single cell sequencing. And I know this is something that's very familiar to you. But I worked for years on bulk sequencing data. And I think the issues with bulk sequencing data were most apparent when we did the TCGA Immune Landscape paper. Within that working group, we must have tried over 20 methods. And it was like a 2-year project trying to see whether or not we could deconvolute the TCGA data. In the paper, it is mentioned, but it's not focused on because it's actually a very difficult thing to do. And although we produced gene sets and we did various things in that paper, none of us were terribly happy with those subtypes that we produced. And it kind of really, to me, emphasized the limitations of working with bulk data. Now, I'm just going to briefly mention where we're applying the single cell sequencing data without going into the biology, because given the audience and the various great conversations I've had with each of you, I've realized that this audience is primarily computational, so I'm just going to focus on the methods. But where we've been applying single cell sequencing data is looking at NK cells. So there was a recent one where we looked at natural killer cells. We did both CRISPR and PRISM and a lot of single cell data there. We've used it looking at, and we're developing CAR-NK cells in solid tumours for the first time. These are in blood cancers, but they aren't in solid tumours, and we're hoping to get those into clinical trials. And we've also been looking at minimal residual disease, or those cells that persist in the presence of drug. And these were all papers that were recently published. This one was interesting because there was 3 papers that were co-published together. Unfortunately, ours was bumped down to Cancer Cell. The other 2 were published in Cell. They were all supposed to be co-current, but it's on this epigenetic mechanism that we all found around the same time to be very important in cancer, that is only observed in 3D organoids or in better culture models and not in traditional cell culture. Anyway, so what are the problems when we're doing single cell data analysis? The data we know is sparse and noisy. It's got lots and lots of zeros. It's high dimension. We're looking at tens of thousands and millions of cells. And in many cases, we're dealing with platform variability as well. And we're frequently trying to integrate data from different sites and different studies. And I'm going to talk about those issues. So we put in a grant, we're in year 3 of it, to the CZI, Chan Zuckerberg. And I said, oh, we'll do data integration and we'll do pathway analysis and we'll do all of these things. But once we started looking at it in a lot of depth, I realized we needed to go back to brass tacks. So within single cell pipelines, one of the things that we always do, does this work? Okay, is the first thing we do is we find embeddings for the cells. We represent the cells in a lower dimension. And typically that's done by PCA. Thereafter, so we do some MAPES QC, filtering, normalization, we do the dimension reduction, and thereafter all the downstream analysis happens. And frequently people pass by these first 3 steps using whatever is the default option in the software that you most prefer, and they go straight into the RNA velocity and the clustering and the interpretation of the clustering, and kind of ignore this section here. However, that's exactly what I'm going to focus on. I'm going to say why this is actually critical, and it's really important that we focus on this aspect, because otherwise what we see downstream is potentially erred or confounded. So what is PCA? PCA was first described in 1901. It's over 100 years old by Carl Pearson. Interestingly, and I kind of always quote this, it's worth knowing the history of Spearman and Pearson and all these fantastic statisticians and their relationship with Galton, and the eugenics. I think we all have to stand up and own this as a field. So please, if you don't know that history, go and read it. But what is PCA? It is, you're trying to find linear projections through the data which capture most of the variance in the data. So the first projection through the axis should represent most of the information or most of the noise. If you're thinking of a matrix of RNA-seq, that may be the maximum number of genes that are correlated together. Maybe you've got a couple of 100 genes that are involved in the same pathway. That may well be a source of variance. Or it could be that you've got 2 batches in the data. And we've done multiple kind of reviews in this space. And PCA can be computed by various different ways using eigenanalysis, singular value decomposition, latent factor analysis. And I have a GitHub vignette which basically goes through all of these different computation approaches, all of the different ways that it's implemented in R, and how they all relate to each other. When you get factors out, they're called principal components, principal axes, latent factors, eigenvectors. There's many, many different names, depending on whether you're coming from a computer science, a physics, or a statistics, or even an ecology point of view. So all of these terms are actually frequently used, and people may actually not understand that they're talking about the same thing. And normally with PCA, these eigenvectors or principal components are actually ranked such that the first represents the maximum variance. That is not the case with all of these decomposition methods. For example, if you're using independent component analysis or NMF, they're not. If anybody here has young kids, you know you spend most of your time trying to put structure on the playroom. So this little picture really appealed to the person who spent way too much time trying to tidy the playroom. The idea that you're trying to find these linear vectors through the space that that capture variance in the data, whether it be small trucks, construction trucks, and utility trucks. And thereafter, we're applying all of the lineage reconstruction and the trajectory analysis and everything else that we do. If you don't realize that these are steps in the pathway in your single cell analysis, yes, UMAP, TISNE, all of these include a PCA step or expect a PCA step. In 2019, the University of Michigan study compared various different methods, and they looked at 19 different methods for dimension reduction that had been proposed at the time. And this included methods like some of the negative binomial methods and zero inflated methods. And partially because of speed and partially because of the results they observed, actually PCA and factor analysis described in 1901 and described in 1904 were the methods that they said outperformed all other methods. I'll talk a lot about GLM-PCA here, which is widely used. And some of the problems with these methods is that they're iterative and you're not guaranteed a solution each time. And they're also computationally expensive methods. But whatever of these downstream methods, you're probably doing dimension reduction at the start. So we went back to really basics and we said, what happens when you do a PCA on single cell analysis? And we published this little review of it. So what most people do is they'll do some sort of a transform on the data, and then you're doing a PCA on that. And those transforms, maybe I can use the mouse, can I? Yeah. So if you're given some pretty normal looking distributions here and you do these transforms, you get reasonable results. If you've got some simulated 10x counts from single cell data where it's really sparse and you've got a lot of zeros, most of these transforms do not generate something that looks any way normally distributed or quantitative the PCA expects. So scaling by a factor, that's typically done to correct for library size in single cell RNA-seq. Centering of the data, this is part of forms of PCA. Standardizing the data to a z-score, subtracting the mean, dividing by the standard deviation, or some other transform, like a log transform. These are all methods that are used in that pre-processing step that people use. But as you'll see, we rarely get something that looks like a nice bell curve. So what exactly is most PCAs doing? So there's two versions of it. There's covariance-based, where you just column center the data. You subtract the column center mean, and then you do a SVD, a singular value decomposition. Or you've got a correlation-based PCA, which you subtract the mean, you divide by the standard deviation, you get z-scores, and then you do an SVD. This is the R code to compute an SVD from scratch. We're basically scaling, we're centering, we're dividing by the standard deviation, and then we're running SVD. You can compare the results of this to any output from any package you're doing. That's exactly what a PCA is doing, nothing more. So we're taking the original data, we're getting the z-scores, we're running the SVD. SVD, singular value decomposition, gives us three matrices, the U, the D, and the V. The D matrix is the eigenvalues, which tells you how much variance is actually represented in each eigenvector. The U and the V are basically the scores associated with the samples and the variables, or the columns and the rows. Okay, what happens when we do this and we actually look at this first step that is so widely used in single-cell RNA-seq? Actually, it's not very good. This is benchmarking data from Matt Ritchie's group in Australia. And this is a very simple study. It's 3 cell lines that were performed on 3 different platforms. They looked at CellSeq, 10x, and DropSeq. And what they did is they took 3 cell lines, so 3 platforms, these colors, blue, green, and orange, and three different cell lines, mix them together. So these cell lines should be very different, and we'd want to see no platform differences. When we actually do this, this is what we see. So typically people will do log counts, or they may do raw counts, and there's debates about that in the field, and I'll go into that in a little bit for a second. But what we see when we do a PCA, in raw counts, we see this really weird horseshoe shapes. What do I mean by a horseshoe shape? Well, one of these typically is the first PC, which is the horizontal one. You see that the data are skewed to one side of the axis, and you're getting this horseshoe shape. Okay? On the other side, if we log the data and we don't do any pre-processing, we get really bad results. But even when we do the pre-processing, like the centering or the z-scoring, we see whilst we can separate the cancer cell lines, we can't separate. The platform effect is very evident. So if you see here, the 10x are actually going to the outside here, and the DropSeq and the CellSeq are actually here in the middle. So this is a really simple study. We shouldn't be seeing this platform effect, but it's not doing a great job. So I mentioned horseshoes. Do people know what that is in PCA? No? Okay. So it's something that's well described in the PCA literature, and it's something that everybody, whoever runs a PCA, should be aware of. This was like 2018, and it was when I started playing around properly with single-cell RNA-seq. The first thing I did was I went, oh, I'm seeing horseshoes. And of course, you know, being whatever, I decided to post on Twitter, and this was like a little analysis I ran here, and I'm seeing all of these beautiful horseshoes here all over the place. And then we started a whole discussion with various other people saying, oh, we get horseshoes in microbiome data, we get horseshoes in metabolite data, we get horseshoes in all of these zero inflated data sets. We see it in some of the PCA and genetic variation, and yet people don't actually recognize that this is an artifact in PCA. And it's very easy to recreate this. This is actually from Morton et al. Paper, who actually responded. Jamie Morton was actually on this Twitter thread where we had a big discussion about horseshoes. And what you can actually generate it. So if you have this data here, where you have something that's a gradient through the data. So in this case here, all of the zeros are white and the actual values are black. You get this amazingly beautiful PCA here. Perfect one. And Susan Holmes from Stanford has also published on horseshoes and PCA, looking at the voting patterns of Democrats and Democrats and Republicans in the US House, and she's actually simulated data even to get S shapes. So you can actually get these really nice shapes once you know that this is just bias in the data. So it's something to be aware of. And even when we don't have the horseshoes, we actually can explain why we're seeing this lib size effect. So if we actually correct for this, we can actually remove it. And the way that it's recommended in a lot of the manuals is to do this really complicated linear regression where you're regressing out the library size. And I'll tell you why that we can do it much simpler and easier. But this basically is those PCAs. This is library size, the depth. And you can see here that the 10x data had a higher library depth. And you can see that you can see these artifacts in the PCA. It's not adjusting for it. So all of these things are known in the PCA space for a very long time. In fact, we did a review way back in the pre-single cell data space of using dimension reduction methods. And it was in 2015. We got published 2016. And we actually state the limitations of PCA and why it doesn't like zero data. Unlike PCA, correspondence analysis, which is another method I'm going to describe, can be applied to sparse count data with many zeros. So this is not something that's unique, it's well known, but yet in the single cell space, everybody's ran away with just doing this log transform centering and SVD approach. Yeah.
I really don't understand. So let's say I have a lot of zeros and I get a horseshoe. And why is it a problem?
Because what people tend to do in a lot of it, particularly in machine learning, they'll take the first 10 components and you'll put it into a machine learning algorithm. When you have that horseshoe, that first component normally represents a lot of the variance. So it will actually get a lot of bias in any machine learning algorithm. And that first component literally just represents structure in the data. It doesn't represent any biology. It doesn't represent anything that's useful. So you're kind of losing one of your components. Like components, potentially components 2, maybe another zero, another artifact. But once you get on to 3, 4, 5, and 6, you will have information there. But those will have lower variance and they'll have lower weight in your actual analysis. So you're putting in a lot of noise into your model.
So the first component, you said then captures structure in the data, and the structure is about zeros and non-zeros?
It's about zeros and non-zeros. You'll also see it if you put in anything that's got scale. So in PCA, you're visualizing data in Euclidean space. So if there's a scale or a distance effect, you'll also see that in the first component all the time as well. And it's not useful information by and large where we're trying to find like correlations and connections. The fact that you just have a scale in the data is not normally something that's useful, and you're putting in that uninformative principal components into your learning model.
Sorry, what are you calling scaling the data? So
the classical one, if you do like height and weight, you'll always get like this weird first component because there's a big scale normally there between those two.
What is the scale here?
So the fact that you've got so basically PCA expects a nice bell-shaped quantitative data. It really hates zeros. It doesn't like them at all. It doesn't like multimodal data. It doesn't like kind of unimodal data. It doesn't like skewed data. It just doesn't behave very well. And you're looking in that Euclidean space. Like I often say to people, would you use a Euclidean distance to look at your data if you were doing a hierarchical clustering tree? And most people will say, no, I'd use correlation. And then I go, why are you using PCA, which is looking in Euclidean space? Now, if you transform the data such that the distances you're looking at are more informative, which is what I'm going to describe, then it's completely appropriate to z-score normalize an SVD if you want. But it's just that first step that people often just miss. That's actually, you're throwing a lot of information into your t-SNE plot and your cluster calling that you don't need to be putting that in. I'll describe in a bit more detail. So there's lots and lots of different methods to transform data into a more useful singular value decomposition. So if you're doing, a lot of the times people have genetic distances or they have binary data. And there you can use a binary distance metric and you can use something like a peak away plot, which is basically a decomposition of a distance matrix. I'm going to describe correspondence analysis, which is decomposition of the Pearson residuals. As I said, principal component analysis is decomposition of the z-score matrix. So all of these methods, you're slightly changing what distance you're looking at in the data. Now, correspondence analysis, which was one of the ones in that review that we said might be useful, was described way back in 1935. It was very popular in the French School of Statistics and Benzacry School in the 1970s and 80s. It was taken up by Michael Greenacre. And also it's like in like basic things reported by Tukey in sort of like Tukey's books. These are all described here. This is very widely used in things like ecology and actually in some of the marketing things as well, where we're looking at count data. What is it? It's simply a decomposition of the Pearson residuals. So here, your observed are your actual values. The expected is basically a product of the row and the column weight. So if you sum up the row values, you can get row weights. If you sum up the columns, you can get column weights. Take a product of that, that's the expected, and that will actually give you then the Pearson residuals, and you decompose that, and that's what correspondence analysis is. So essentially then you're looking at the strength of association or the chi-squared statistic between the rows and the columns. So in a way, it's actually much more closer to looking at correlation in a hierarchical cluster tree. Whereas PCA is always going to be kind of like decomposing in the Euclidean space. And this was being recognized in the field. So Will Towns, who actually was at the Farber at the time, we were having a lot of discussions within the Farber, and Harul and Sajay actually were telling me that they had actually looked at my review from 2016 as well, and we were recognizing that, you know, we need to be not looking at the logged counts, we need to be looking at the Pearson residuals. And what happens if you just do this simple transform on the data? Well, up in the top here is the PCA space, and here is the correspondence analysis space. So this is the decomposition of the Pearson residuals. Whether you look at counts or log counts, you actually see that you immediately remove that batch effect, that library sequencing depth. This is a really, really fast method to compute. It's equivalent to PCA, so you're actually not getting any more expense with time. It works very well. Okay. So one of the things that people were starting to do before this conversation started was actually throwing canonical correlations at it. So within Seurat and within Harmony is another method. What people were doing is we're getting these batch effects in the data with the library sequencing size. Well, let's throw canonical correlations at it, and that will actually align our batches. Actually, it doesn't. So this is taking that same data sets where we have two principal components that we're running, and we're like basically aligning with canonical correlations analysis. And as you can see, this is, first of all, many steps, so it's more complex, it's more computationally expensive, and it's a worse result. So if you have PCA and you're seeing this library batch effect and you're trying to do Harmony or you're trying to do canonical correlations analysis to align these, you're not fixing the problem. You're not looking at the Pearson residuals, you're still looking at the logged counts. You still won't get the result that you want, even if you try and force it. And, you know, I think this is kind of established in the field, but we did like comparisons of various different methods. So we looked at the SC mixed data set, we looked at the Zeng mixed data set, we looked at various different single cell benchmarking data sets. We compared principal component analysis and using our implementation of correspondence analysis. So we've implemented this with like kind of sparse SVD behind it, Herbala, so it can run a little bit faster. We've implemented multiple different iterations of SVD behind it, and we've actually implemented it so it kind of maximizes the matrix stats programs which are designed for dealing with sparse data. And we've implemented in a package called Coral. So Coral here is doing decomposition of the Pearson residuals or correspondence analysis. This is PCA. We're looking how well we're doing. The green here is correspondence analysis. The purple is PCA. The closer to 1, the better you're recovering your clusters. And you can see in almost all cases, we're doing better with correspondence analysis. It's not always the case. So where you've got like a very simple data set, PCA will still do pretty good. But, you know, we actually see improvements with correspondence analysis. And this is one of those data sets. This is the Zeng Mix 4. This one here. And this is just a visualization of it. So here is whether we do the counts or the log counts, and whether we do correspondence analysis or PCA. If we do PCA on the raw counts, we get the beautiful horseshoe. If we do it on the log counts, we're getting nice separation. And both with correspondence analysis, we don't have to log the data. And there's a really nice review by Will Towns saying when you actually log single cell RNA-seq data, you actually introduce bias when you actually log it. So there's problems with this log +1. So anyway, but there's problems with correspondence analysis. And don't let anybody say that, you know, just doing decomposition of the Pearson residuals is what we should be doing. And that's kind of the best that most of the field has got to at the moment. And we're saying, no, it's actually not good enough. We know that correspondence analysis is sensitive to outliers. And this is a plot that we did, and we're looking at pancreas cells here. And if you look at islet cells that just secrete tons of insulin, or you see this as well if you've got B cells that are secreting lots of antibodies, you see these cases where if you plot the results of a correspondence analysis with dimension 1 and dimension 2 here, that these little outlier cells just drive it. And that's expected in these data. We expect that secretory cells will be producing a very high amount of a protein. And because they're the only cells that are producing high amounts of that protein, if you think of it, the observed minus the expected, yes, that's no other cell is producing insulin, and that cell is producing tons of it. It will have a very high chi-squared statistic, and it will skew the entire plot. So this is to be expected. Now, some of the ways people have started to look at this within Laus et al. Recently published a paper where they're trying to trim the data. Within Seurat, they're trying to do trimming things, but some of these are just kind of a little bit arbitrary and ad hoc approaches. So we said, well, actually, let's look at this properly, because we know that the decomposition of the Pearson residuals using a traditional correspondence analysis approach is actually very sensitive to outliers, and we expect to see lots of overdispersion in our RNA-seq data. So how do we look at this? Okay. So, and this is all work by she was a master's student who kind of then has stuck with me a little bit longer. This is all work by Lauren Hsu, who's currently undertaking a PhD in Harvard School of Public Health. So what we compared was 3 different approaches. So one, change what chi-square statistic we're going to use, how we compute the residual. The second one was use a variance stabilizing before we apply correspondence analysis. And this was kind of described by the Laus et al. Paper. Or the second one was apply smoothing residuals, which happens in Seurat, in the SC transform 2. So the different methods, standard correspondence analysis. This little method here, using the Freeman-Tukey residuals, wasn't something we knew anything about, but actually has been applied in archaeology. So if you're an archaeologist, you go to these sites and you collect artifacts. And those artifacts are really sparse. There's lots of zeros because these artifacts are only seen in a couple of sites. But you may have many different sites and you're trying to integrate these artifacts by sites to try to get some information about your archaeological digs. And so we actually found in that space. We looked at square root transformation with correspondence analysis, Amscoon transformation, and correspondence analysis with the Freeman-Tukey count transformation. All of the equations are here. And we also looked at a power deflation. And so how do they each do? We applied these to 9 different data sets that are kind of used in the field. So the PBMC data sets, the pancreas data sets, the brain data sets. And we wanted to do something with more cells. So we looked at the Xenopus tail data set. And so this is these results. So these are our 3 different approaches. Correspondence analysis with different residuals, different variance stabilizing approaches, and the smoothing transform. And we compare these to GLM-PCA because that is one of the, it will basically do decomposition on Pearson residuals using this generalized linear model approach. And I'll explain a bit more about that in a second, but it's considered one of the best alternative approaches in the field at the moment. And the data were pretty clear that we should actually be using the Freeman-Tukey residuals. And so we've started in our own group using this approach when we're actually decomposing single-cell RNA-seq. Now, there's a couple of cases here where, a slightly different method, some of these variance stabilizing methods do well, but here they're all 0.888, so they're actually all equal. Here they're both 0.75. There's a little bit here with the Zengmix 4 data set, but it's not, it's marginal. GLM-PCA does better in a couple of cases. It's equivalent in the Zengmix 4. It does better in the brain pancreas, and it does marginally better, 0.67 versus 0.65 here in one of the other brain data sets. But what I'll do is also explain to you why I think these determined methods are actually preferable. First of all, is computational cost. GLM-PCA is an iterative method. It is actually really expensive to run. So this is GLM-PCA. We're looking here at the number of cells, going up to 20,000 cells here, small data sets, and this is the runtime. And just with 15 or 20,000 cells, you're looking at a 40-minute runtime if you want to do GLM-PCA. Whereas all of these methods, what we've done here is we've blown these up, because here we're talking about less than 2 minutes. So there's a big, big difference once you get onto typical size data sets in using some of these iterative methods that are described. This is here the number of features, up to 20,000. And this here is the Freeman-Tukey residuals, and it's actually the fastest. Now, these are marginal times, like this is 1.5 minutes versus 2 minutes, but 1.5 minutes versus 2 minutes does actually expand a bit more when we get into, 1 million cells. So we actually liked the fact that our best method was also our fastest method. Okay. The second thing that happens with some of the iterative methods like GLM-PCA is you're very determined, because it's iterative, you're sometimes dependent on you found a lucky seed. So this is one example here. This is looking at the Lal or pancreas data set. The grey ones here are GLM-PCA, and in this case here, the seed fails to actually find a reasonable decomposition. And sometimes, where's the Xenopus data set here? We see that we've got a lot of noise about which seed it starts with. So again, you may do better with GLM-PCA, but it probably depends which seed you get. So for us, it's like, if we can use a determined method and get a result that's always reproducible, or do we have to iteratively rerun it multiple times to get the best result? So for those reasons, we actually like this approach, and we think it's a reasonable approach that people can be using that can potentially give you better results in single-cell RNA-seq. And we've implemented this within the bioconductor. I do a lot of work within the RNA bioconductor space. Lauren is saying that she'll probably also write it up in Python. We haven't done it yet. Or maybe we have done it, but she hasn't released it. I actually need to ask her. But basically, within the Corral package, we kind of stripped down everything to bare matrix code. We use matrix stats. We use basically all of the sparse implementations. So we can pretty easily, and it's written very nicely modular, so we can push in other SVD approaches should they be described, and we can easily push in other transforms should we define those as well. So it's actually, I was quite happy the way we rewrote it all. So one of the other advantages of correspondence analysis is when you're looking at the strength of association between the rows and the columns, you can now present those rows and those columns in the same decomposed space. This can be kind of nice because you get a really nice intuitive way to pull out features that are actually important in the multivariate space. And so this was, again, from that review that we wrote in 2015. And this was, we have brain, melanoma, leukemia here, and we just have some genes and some samples. And you can look at the genes here on the correlation circle. And anything that's projected in the same direction or the same angle are actually correlated. So let's look at that. For example, if we take these three here, we can see, or maybe I should just use this with the mouse and stop. If we look at the actual values, we're seeing the nice correlation between those three here. And equally, if we look at these here, we can see the nice correlations up here as well. So we have that nice idea of being able to pull out those correlated elements. And the second thing is the greater the weight of these genes in this space, the higher the chi-squared statistic between that gene and that sample. So it's a very intuitive way that we can start visualizing genes and cells in the same space. And so basically this is just in the embedding space here of the genes and the cells. If we have the cells that are the black ones and the genes are the triangles, We can look at that angular distance, which will actually give us that strength of association. And we can look at the magnitude, which is the direction from the origin. So, for example, if we're looking here at PBMC data, and this is 8 cell types in peripheral blood monocyte cells, and we can pull out the genes, we say, that are most associated with regulatory T cells here, or that most associated with the monocytes here. And we can look at these ridge plots, and we can see very clearly that this gene is strongly expressed by the green cells here. Okay, so it provides a nice way to actually visualize some of this. Okay, so basically, the Freeman-Tukey residuals was the most performant. And when we apply these methods and then do downstream clustering, we do better than when we just do standard PCA or correspondence analysis methods. And we've implemented in PCA. Now, we've got, I think, just 5 minutes left. I'm just going to talk a little bit about where we started with all of this, which was the multimodal idea. And that's where I kind of wanted to go with this. All of the code is implemented in Corral, and the paper is up in bioRxiv. So let's just take that one little step and say, right, now that we've sorted out a little bit of the decomposition of one data set, what can we do for multiple data sets? And this is something that in the bulk space I'd worked a lot on, and this was kind of what I did during my postdoc and kind of what we did when I had started my lab as well, was how do we bring different data sets into the same space? And we described lots of different methods for doing it. And again, we kind of reviewed that in that 2016 review. There's a lot of different methods beyond canonical correlations analysis. We looked a lot at multiple co-inertia analysis, where you're maximizing the covariance between the eigenvectors of the data sets in the same space. But there's also various sparse implementations and various ways that you can weight the data sets in space. This was an example that we published, oh my God, in 2014. This was actually multiple co-inertia analysis. It was implemented at the time in the OMICAD paper, or OMICAD package in R. And in this case here, we're looking at 4 different transcriptomics data, 1 proteomics data. Each cell here is visualized by a little star representing the different omics. And if those omics are very consistent, or giving same information across the data sets, we see a little tight star. If one data set has more information, for example, that captures features that are important for those cells, we may see this like long tail here, which in this case represents the proteomics data has more information. These here are leukemia cells. We can see the clustering of the cells here. We can also pull out each of these different, the genes that are most associated with those leukemia cells from each of the 5 different platforms. And we can also do this in multimodal. This was actually described in that 2016 review, where we looked at mRNA, protein, and RNA.
Sorry, can I?
Yeah.
What is behind this integration? So you take these different.
So that paper described multiple co-inertia analysis. So given multiple decompositions, you actually constrain the decompositions so you maximize the squared covariance between the eigenvalues. So when you're trying to find the actual eigenvalues of the individual data sets, you constrain it, and that constraint finds for co-inertia analysis, you're maximizing the squared covariance between the actual eigenvalues. For canonical correlations, you're maximizing the correlation between the eigenvalues.
Okay, that's very, very nice. And I was, can you go back just to so I understand what I see on this?
So this is,
Visualize the difference.
yeah, so these are different cell lines. So there's like whatever, about 5 or 6 leukemia cell lines. There's half a dozen melanoma. There's half a dozen brain. Oops, sorry. And the round circles are the mRNA, the triangles are the proteins, and the miRNA, so the microRNAs are the actual squares. In this CNS cell line, you're seeing a lot of information in the microRNA space. In this melanoma cell line here, you're probably seeing more information in the mRNA space. And we're just visualizing all of the variables in the same space here. And then, so...
But you say different decompositions, right? So you actually decompose.
You actually do a joint decomposition of them all together. You can do the individual decompositions and then you basically rotate the axes. The simplest kind of thing that people find the easiest to understand is kind of Procrustes rotation. So with Procrustes rotation, that's kind of like the granddaddy of all of these methods. With Procrustes rotation, you're just like rotating the axes through such that you get correlation between them. So that's kind of the simplest approach that you can use. And then basically there's various different ways that you can make that more complicated. But all of these can be described by like a simple matrix operation. These methods are like Stefan Drey, Sandrine Dolodek, we've done it, Kim-Ann Lacau. Who else has done a lot in this space? These have been described time and time and time again in applied to bulk data and applied to other data sets as well. So I can give you like a stack of reviews of the space.
Okay, but then I can...
Within MOGSA, which is another R package we did, we actually implemented those so you can actually do many multi-block methods with the same code.
If you are doing the decomposition separately, how does the integration happen?
So if you're doing the integration separately, what you then can do is take the integrations and rotate the axes.
Then you put them in the same space?
Yes, and then they'll come into the same space. But normally what you do with these is we do transformations of the data and then we do these multi-block integrations. So, and there's to kind of get into this would be a whole talk in and of itself, because how they actually work, there's nuances between the different methods. Status is oh my God, I can't remember now what that's what that is in TensorFlow. It's got a different name in TensorFlow, but it's basic CP decomposition, multi-factor analysis, multi-correlational analysis, canonical correlations analysis. They're all very similar if you think of these as matrix operations in multi-data sets in the same space. And so we actually implemented all of these in just one package. But author Tenna House has done a really, really nice review of these as well, because the differences between kind of co-inertia analysis and canonical correlations analysis is really whether you're maximizing on that correlation or covariance. So these methods are actually they're very similar. And you can basically play with how you pre-process the rows and whether you weight the data sets. And weighting the data sets is something that's a whole field that we haven't even begun to explore. What we did in single cell space is something that's ridiculously simple for the moment, because this is something we're going to get into, but we haven't got into yet. What we did is we implemented this Corral-M, which is really, really simple. We're actually just using the standardized residual matrix. We're concatenating it and we're decomposing it. It's not even a proper joint decomposition, but actually it seems to work quite well. And we compared it to using Harmony and Seurat. Seurat uses CCA, and Harmony does PCA, and then it does a soft iterative clustering. So it kind of does this it does PCA, and then it does iterative clustering. Oops, I just done something. Yeah. And okay, here's the result on that. Basically, we don't do quite as well with Seurat, but we're way faster. But we recommend that you can use Corral-M as an input into Harmony and you can get way better results because we're using these Pearson residuals rather than the PCA space. So we don't do as well as SC transform, but we actually are way faster. Okay. And this is basically looking at PBMC data, comparing Harmony, Seurat, then comparing Chorale, looking at different cell types here. Okay, so we're just a lot faster. Okay, and just a plug for the Beers method that we did in 2020. If you're interested in this data integration space, we did a hackathon. It ended up being online. We looked at data integration of multiple different problems, and all of the code is on GitHub. So all of the methods that we and other people did to try to integrate all of this data set, all of the code is there. There was like 30 different submissions where people tried to integrate these data sets. We were working with Bioconductor. These are links here to kind of ways to do things. So in summary, we need to move beyond just z-score normalizing and singular value decomposition, which is PCA. We need to think what are the appropriate transforms for the data before we apply singular value decomposition. There's many different data processing approaches which can result in a better decomposition, which are better for data which have zeros and counts and gradients. We may do better than CCA for joint decompositions. We need to look at that. We need to look at how the data are normalized and weighted in the joint decomposition space. And I didn't get into all of this, but like the MOGSA package and like a lot of the other work we've done is projecting annotation into that space, so then you can get pathway scores and gene set scores that I didn't begin to get into. And I just want to thank the various different people that I'm working with.
Thank you. Really beautiful talk. So if you have any questions for Aedin, because of the sake of time, we can. We're running really late.
I've got jobs.
Yeah, computer. Maybe you can ask a question.
Do you have any idea how we can apply this correspondence analysis and this integration to integrate features from histology and omics data? And somehow, of course, the data we have in omics and the phenotypes we see in the histology images are somehow related because the proteins determine all this density and morphological features. But if we somehow manage to remove like the features which are not independent within these two sets and somehow integrate it.
So the first thing is that more than likely the data that you have from the histology space will actually be much more normally distributed. You normally see the PCA as appropriate to apply to those. So you can do a PCA on that data, you can do correspondence analysis. It's kind of what transforms you use and how you actually look at the data. Or you can do sort of like a correspondence analysis where doing the chi-squared statistics on both. Within the space for where you're doing the spatial analysis, what we did when we did that Beers analysis is we decomposed the Moran index and we did a Dialongi triangulation of that to actually look for clusters in the space. And all that code is on that GitHub. And other people kind of did similar kind of approaches to that. So what you're decomposing in the spatial space is actually the spatial kind of co-clusters that you're looking at. And then you can do those. If you look at that paper that we wrote on the different methods, and if you look at that GitHub, all of those methods are there. And I'm happy to show you and email you and help you with it.